Hello again. We're ready to begin lecture number 8. I hope you have listened to the other 7. This movie will cover the identification of biological fluid stains at crime scenes. I will reserve the individualization lecture for next time. So we will talk about how you identify a stain at the crime scene. The most common stain that you find, of course, is blood, because it's red and it's easy to see, and we all associate that with violence, and so we're always looking for blood. And so let's talk a little bit about blood. It's an extremely complex mixture of nutrients, antibodies, cells, and the one thing that we can identify in blood is hemoglobin. That's what makes blood red. Most mammals have hemoglobin that is slightly different from human, but basically the same, performs the same function. So if you come upon what you suspect is a blood stain, we don't assume it is. We're going to test it. And we've had what we call presumptive tests for blood for many, many years. The original one was benzidine. Turns out that hemoglobin is what we call a peroxidase, meaning that it helps to facilitate reactions with peroxide. There are other peroxidases in your body, but hemoglobin is a primary one. So if you have a reaction that requires oxygen and changes color, then you can use it as a test for blood. The first one was called the benzidine test, because benzidine was the initial reactor that you put on the sample. So you would take a sample of blood, and it could be very, very small. This is a very, very sensitive test. And you would add peroxide. Hydrogen peroxide works. Other peroxides also work. But normally, since hydrogen peroxide is readily available, you would add a little hydrogen peroxide. And then you would add benzidine. If there was hemoglobin present, the reaction would take place. Hemoglobin would facilitate the reaction. We don't think that the hemoglobin is significantly changed. That's why it's a peroxidase, because it facilitates reactions but doesn't really involve itself in the final product. So the reaction would take place, and the resulting material would turn a greenish blue color. We used benzidine for many, many years, until about in the late 60s, early 70s, it was discovered that benzidine is a carcinogen, meaning that it causes cancer. In this case, quite often cancer of the bladder. I have a couple of friends who got bladder cancer. So we've stopped using benzidine. In the meantime, we have found other equally efficient materials that we have substituted for benzidine. One of those is phenolphthalein, which turns pink instead of blue-green. There are others that also work. So you can purchase these supplies, or you can mix them up yourself. Or what I consider a really good alternative is a product known as Hemastix. This is a bottle of Hemastix. It's very convenient. You can carry this bottle around and it's not terribly expensive. Inside the bottle are these small plastic strips which have on the end of them a yellow pad that has been impregnated with a source of oxygen. It's not hydrogen peroxide, but and also a reactant. It's not phenolphthalein or benzidine, but it works just fine. So I'll show you how this works. So I have a small blood stain here, a lead blood stain here, and what I'm going to do is I'm going to rub it with this piece of filter paper. I'm not going to rub it completely off, just going to rub the blood stain with the paper, and hopefully enough hemoglobin will transfer to get a positive test. It's a very small stain and it's on a cotton cloth, so sometimes it doesn't transfer very well, but we'll see how it goes. So I've just rubbed that. You can see that the stain is still there. We just hope there's enough that's been transferred onto the paper. So I'm going to get the paper a little bit wet. There's two ways we can do it. Maybe we ought to get the hemistick ready. So here's the hemistick. Nice little plastic strip with the chemicals embedded in a pad on the front. That yellow pad has the chemicals impregnated into it. This is a test that was originally designed and still used to test for blood in a urine sample. So it's used by doctors to test to see if you have blood in your urine. You're not supposed to have blood, or at least not supposed to have very much blood. Some people have a little bit, but anyways, so we've just adopted it. I'm going to get the hemistick a little bit wet on the pad. You should use distilled water, but other deionized or regular tap water will work. As chemists, we prefer to use distilled water for everything we do. So now I've just touched the filter paper with the hemistick, and you can see how it's beginning to turn green. And the tip of the hemistick is also beginning to turn green, so that's a positive test. Easily done. You should, of course, save the hemistick in a plastic bag and record the results with your camera. But it's a great little test. You can see that it's very sensitive and doesn't destroy the stain to begin with. It doesn't interfere with subsequent tests. It doesn't interfere with DNA analysis. It's easy and cheap and quite effective and quite economical as well. It has the advantage over phenolphthalein, which is the name for the phenolphthalein test is Kastle-Meyer, and a lot of people like Kastle-Meyer, and I like it. It's good, but it's 2 bottles of liquid that you have to carry around. With the hemistick, all you need is a little vial of water, and as I say, it's dry and it's easily done. So I like hemisticks. I should hasten to say that it's a test for hemoglobin. It's not a test for a specific type of hemoglobin. So it'll work with duck blood, it'll work with deer blood, it'll work with cat blood, it'll work with dog blood. Any blood that's red, it will work with. And there are other peroxidases in the world. Some of them are in plants. And so it's not an absolute test for blood. The peroxidases that are known, however, none of them are red. None of them look like blood. So I suspect that if you were at a crime scene and you saw a white stain, you wouldn't think, oh, I wonder if that's blood. You could test it, but the plant peroxidases are usually much slower and weaker. But nevertheless, if you get a positive peroxidase test, what you should say is, this stain tested positive for blood. You shouldn't say this is blood. You say it's tested positive for blood. And that's absolutely correct. And there are virtually no known materials that look like blood that test the same way as blood does. So I'm not a doubting Thomas in this case. I like this test and I use it a lot. And it's particularly effective if it's a negative. That is, if it doesn't turn green or pink, whichever test you're using, you can say this tested negative for blood, and the odds that it is blood are very, very remote. So it's a great test and a preliminary test, or what we call a presumptive test, which requires that we do additional testing. Another peroxidase test is one called luminol. Luminol has been around a long, long time, and luminol is a compound that glows in the dark, luminesces. It luminesces a pale blue color, and lots of people get excited about it because it glows in the dark. So that's a neat test, right? Well, it is a good test, but there are a number of things that give you false positives, metals, certain metals, and other peroxidases. So I reserve the luminol for situations where large areas need to be covered. For example, if you think that there's blood that's been cleaned up on a carpet, for example, and you couldn't use the hemistick because it's too big, you'd have to use 100 hemosticks to touch every part of the carpet. So you just have to darken the room. Usually we just wait till it gets dark, but if you're in a big hurry, you can put black plastic over the windows or whatever. Anyway, you darken the room, and then you spray the liquid luminol solution, and almost immediately you'll see a blue luminescence if there's blood there, and it will show you blood that you haven't been able to see. That's the idea of luminol, is if you have blood that you can't see because it's in such trace amounts, and it's over an area and you're not exactly sure where it is, then luminol is a good alternative. There's a new material out called Blue Star, which luminesces even brighter, and is available, and some laboratories have used Blue Star with success. But these are, again, reserved for special situations. If you've got blood on the wall, it's red, and you got spots and you can see them, then you shouldn't use luminol for more than one reason. One is that hemosticks and Kastle-Meyer are quicker and easier to use. Second of all, with luminol, you're spraying chemicals in the air which are not necessarily good for you. And third, it has to be dark. I guess one other issue is photographing it, and you have to take some special steps to photograph luminescent materials, and you're doing it in the dark. And so all in all, I like luminol, but only in those situations where it's absolutely called for. We had, for example, we had a woman down at BYU who was found on her bed with a bullet hole in her head and a gun right by her. Fortunately, the investigator was a Weber State graduate. By the way, this is the only homicide I've ever investigated down at BYU, but so much for that. Anyway, the investigators, as they looked around the house, they found what appeared to be a bullet hole in the kitchen. The kitchen was down the hall and around the corner from the bedroom where the body was found. So there's no way that if that was a bullet hole that she could have been shot in the kitchen and then walked into the bedroom and you know, fallen down on the bed. So we suspected that she had in fact been shot in the kitchen and her body moved to the bed. And we figured, well, you know, she wasn't a huge woman, but she wasn't small either. So we thought, well, maybe she's been dragged across the carpet. So we waited until dark and sprayed the carpet with luminol, and sure enough, we saw a nice broad stripe where the luminescence occurred, and could show that in fact her body had been dragged along the carpet into the bedroom. So they investigated, and the next day a body was found in a car in Kearns with a note saying how sorry he was that he had killed his girlfriend. And he had been the prime suspect, of course, but then we had the note, and so the crime was solved ultimately. And so that's a great use for luminol. It worked in that case, and it's worked in many other cases. But again, it has that specific niche that is, you know, if you have those circumstances, use luminol, but don't think of it as a first resort. Think of it as a way down the line resort. If your stain gives you a positive peroxidase result, the next question is, well, is it human blood? Because as I mentioned earlier, the hemostix and Kastelmeyer do not distinguish one species from another. So there are several tests for species identification. The old test is based on antibody-antigen reactions. Now, you've probably been hearing a little bit about antibodies if you've been listening to the news, and all you hear about is COVID-19, and you've heard it said that maybe some people are going to be immune to COVID-19 because they've build up immunity in their system. Well, you have this very complicated and sophisticated and wonderful defense mechanism in your body. You're constantly being bombarded by invaders, bacteria and yeasts and fungi and viruses. They're in the air, you breathe them in, they're in everything you contact has them, you can't see them. But your body has a mechanism that allows them to recognize most invaders, and so you have to have some way of protecting yourself and not being killed by every little bacterium that wants to eat you. So the mechanism is that your body first of all recognizes it as an invader, and then mobilizes its defenses. And there are numerous defenses, white blood cells and so forth. But one really good one is that your body will recognize an invader and investigate the chemical makeup of the invader, and it will build an antibody to that specific invader. And it takes a few days, but eventually your body will build up enough antibodies that in most cases it will defeat the invader and get rid of it. If it's a new invader, then it takes a while because they have to build a new antibody. But if it's an old invader that's been there before, then you may still have a few antibodies floating around. But even if you don't, then your body knows how to build that particular antibody, and it will very quickly mobilize and produce antibodies. So that's not something just humans have. Most organisms have a similar mechanism. So we can take advantage of that by producing antibodies against human blood. And we do that by letting an animal produce antibodies. And the typical animal in this case is a rabbit. So you take a rabbit and you inject a small amount of human blood, usually just the serum, into the rabbit over a period of about a month. And the rabbit will build anti-human blood antibodies in its blood. So then you can take the blood out of the rabbit. You don't have to kill it. You can just take some blood out and spin off the cells, and the serum will contain anti-human antibodies. So then if you suspect the sample is human, you need to react it with the rabbit anti-human serum. And you can do that in a test tube. Take a small test tube and put a layer of the suspected human blood that's dissolved in the bottom of the test tube, and add a second layer very carefully of the rabbit anti-human serum. And where they meet, the antibodies will react with the antigens. An antigen is simply something that stimulates the production of an antibody. So the antibody will react with the antigen, and you'll see a small white line at the point of interface between the serum of the rabbit and the suspected human blood, if it's human. And that's quite an easy test and doesn't take very long, and we've used it for a long, long time. Subsequently, we've developed some additional tests. The most elegant ones are when we produce monoclonal antibodies, and we can then react them and get a color change. The most well-known one is in what's called a hemotrace card or an OBTI card, and this is what they look like. Just a little plastic card. So, and in the card, whoa, nice. So there's another one. So in the card, you can see on this end there's a little slot, and up here you can see two lines. Well, when you get the card out of the container, it doesn't have any lines on it. So what you do is you dissolve a little bit of the stain and drop a drop or two in this end, and then it will migrate up there's a paper inside. It will migrate up the paper just like thin layer chromatography or any other chromatography. And if the test is working, the C line will show up. If it's a positive test, the T line will also show up. So you can see that that's a positive test. The test is working, and it is testing positive for human blood. And these are easily used on scene. The only problem with them is they're a little bit expensive. So you won't use them as frequently as you would use your hemostick. But they have the advantage of only reacting with human, because they've been manufactured to have an antibody-antigen reaction only to the antibody of human blood. And they've been tested and verified and certified. I have found a thing or two that they will give false positives for, one of which is Coca-Cola. But if you've tested originally with the peroxidase test and got a positive, you don't get a positive from Coca-Cola. You won't get a positive from the things that might give you a positive. And so if you get a positive peroxidase test, you know it's blood, or you're pretty sure it's blood. And then if you do the hemotrace card, then that tells you this is human blood, and that's quite a reliable test and quite popular and easy to do, as you can see, and easy to interpret. Again, you would save that little hemotrace card, photograph it, and save it for for your trial or whatever, whenever you need it. So now we've decided that it's not only blood, but it's human blood, and very little controversy surrounding those hemotrace cards. Everybody likes them. Everybody that can afford them uses them. Interestingly enough, the mechanism for producing anti-human antibodies works just fine for other animals. You can take the blood of a horse or a dog or a deer and inject that into a rabbit, or maybe you want to use a different host. Maybe you want to use a goat, but you inject the blood of the target species into the animal that is going to produce the antibodies. Therefore, you could make anti-deer antibodies, anti-elk antibodies, anti-moose antibodies, anti-alligator antibodies, whatever kind of antibodies you wanted. For some period of time in the old days, we had a little rabbit pen upstairs that we used to make some antiserum, because we did a lot of non-human blood testing for the Division of Wildlife Resources. In any event, then you can use that in the same way that you use the original test that I discussed, where you produce the line in the test tube. A more efficient and certainly more elegant way is to take a petri dish. Into the petri dish, which I show here, you put a layer of agar, very precise agar called agarose, and then you punch holes in the agar, and into the holes you put your antiserum and antibody and antigen. So if you had, say, like 5 or 6 samples that you want to test, you would put the antibody, the serum, in the middle hole here, and the suspected animal blood, that is the blood from the scene, dissolved. You put that in the others. So if you look very carefully at the center hole and the hole on the at about 1 o'clock, you will see a very thin white line in between those two, and that white line is an antibody-antigen reaction, and so the antibody is reacting with the antigen. You know that it's anti-deer antibody, so if it's reacting, it must be deer blood. That way you can test several samples at once. It does take overnight, so it's a little bit longer to get your results, but you can test a number of samples, or you can test a number. If you've got an unknown, you put the unknown in the middle, the blood from the scene in the middle, and in each of the holes you put a different antiserum. You put anti-deer in one, anti-elk in one, and label them appropriately. And then you come back in, and if it's deer, it'll be between the deer and the anti-deer, and if it's elk, it'll be the elk. The elk blood will develop. So this gives you the opportunity to test multiple samples at once and to test against various animals. So I've used this dozens and dozens of times, and I really like it. As noted in your book, you can speed up this process by taking a plate and putting the samples, put a thin layer of agar on the plate, and then put that in a direct current and move the two samples together, because one will be negatively charged and the other will be positively charged. And so that, if you recall when we were talking about chromatography, I mentioned electrophoresis. And so electrophoresis can be used to speed up the diffusion through the gel and get the results in half an hour instead of overnight. But you have to have the proper equipment to do the electrophoresis. Many labs don't. The big disadvantage, of course, to this technique is that you may not have the endoserum for the particular organism in question. We had a situation where a person called the Division of Wildlife and said that she had just witnessed her neighbor kill a seagull. Seagulls are the state bird of Utah, and whether we like them or not is irrelevant. It's illegal to kill them. So they sent out an officer, from a wildlife officer, who knocked on the door of the home where the alleged seagull-icide occurred. And the mother was very defensive and said that her little boy would never do such a thing. And so the game warden said, well, you mind if I look around your yard? No, go ahead. So he looked around the yard and he didn't find any dead seagulls, but he did find a rock with some blood on it. So he brought us the rock. You can't commercially buy anti-seagull antibody. So we told him, well, we don't have access to any standard. We haven't got anything to test for seagull blood. So he arranged to have a dead seagull brought to us, and I didn't question where he got it. I'm sure he did it legally. In any event, we took some blood from the seagull and put it in our rabbit, and our wonderful little rabbit made anti-seagull antibodies, which we proceeded to test against the blood on the rock, and it came up seagull blood. And so the young man got in a lot of trouble for killing seagulls in his backyard. I don't know exactly what punishment he received, but more than he would have received if his mother had been a little more cooperative, I'm sure of that. But in any event, it's an example of how we use the system to determine the allegation was true. We also, of course, recognize differences in the genetic material of different species, and now we have developed techniques for identifying species using DNA. They are very sophisticated and very accurate. They can identify virtually any species. In fact, biologists now use DNA differences to determine if an animal is a particular species or not. If you have two organisms that are very similar in appearance and habitat and so forth, and we think, well, this is two species or one, then we use DNA to help us figure that out. That's another story for another time. DNA will be covered in the next lecture, so we won't talk about the details at this point, but know that we can identify species utilizing DNA if we have the proper DNA laboratory. So next, let's move on to a second relatively common stain that we find at crime scenes, and that is semen. Semen is, of course, very commonly found in sexual assaults. It's also less common, but also found in other kinds of crimes. We had the public defender's office burglarized a number of years ago, and when they attorneys arrived in the morning, there were files, manila envelopes, manila folders scattered all around their office, and they happened to notice that on one of them there was a stain, and they didn't know what it was, so they brought it up to us, and we analyzed it, and it turned out to be a semen stain. I don't have an explanation for why it was there, but it was. So don't overlook the possibility of a semen stain being present in a non-sexual assault situation. Burglars have been known to break into homes, and the excitement of breaking in creates a semen sample, and so you occasionally will find those in that kind of situation. In any event, how do we identify it? First of all, the first thing that you look for is a whitish, crusty stain, and so you have the digital exam or the tactile exam, where you touch it and see if it meets those criteria. Doesn't always, but if it's a fairly pure stain, as it dries, it will dry crusty and white. A second way is to illuminate it with a UV light or an ALS. Semen fluoresces naturally, a pale blue. Of course, lots of other things fluoresce pale blue, so that's not an identification necessarily, but it helps you locate. Say if you've got a bed sheet or a piece of furniture that you have to examine and you don't know exactly where the stain is, then you can illuminate it with the UV light, and when you see a stain that's suspiciously blue, then you can mark that stain and come back and collect it and continue to identify it in ways that we'll talk about later. As far as sexual assault evidence from a victim in particular, you need to consult with whoever the examiner is and instruct them as to what they should do in the collection process, how they should collect it and preserve it, and not ever to cast aspersions on a doctor, but not all doctors are the same. Some of them would be familiar with procedures and some would not, so you have to make sure you talk it over with them and make sure they understand how you want and what you want. You of course should have interviewed the victim prior to going to the medical person for examination, and the victim will tell you, the places where you should look. Obviously, vaginal exam is necessary, but occasionally you need to do an oral exam, occasionally anal exams, occasionally just a body part. So you instruct the medical officer, if he or she finds what appears to be a suspicious stain, to dissolve it in either saline solution or distilled water. Distilled water is preferred, and then just put it in a small container and label it and give it to you. The best way to identify a semen stain is to find a sperm cell. I'm sure that most of you have seen photographs or videos of sperm cells, and that's a human sperm cell. And so they're quite recognizable. They have that oval-shaped head and that long tail, and this one has been stained with a particular type of stain that changes the cellular material's color, but that's the appearance of it. So you should ask the doctor if he's familiar with what they look like, and if so, then tell him to go ahead and make a slide. And he should make a slide with what we call a smear. In other words, you take a swab and moisten the swab with the stain, and then swab that across the microscope slide. But don't let him throw the swab away, because we want to do additional testing on it. If he wants to verify that there's sperm cells, if he's competent to do that, then let him do it. Inform him that he'll probably be called to testify in regard to that. So he or she may or may not want to testify, and so they might decline. But you shouldn't assume that they know how to recognize a sperm cell. Not all doctors look through the microscopes very often, so some of them simply haven't had the experience. In any event, they should make sure that whatever samples they take are air dried. The swab is air dried before it's packaged, and that will keep it from decomposing and deteriorating and not being worth testing for. So then they can do the examination. If they find the sperm cell, then it's identified as semen. There are, of course, other animals produce sperm, but none that I've seen look just like human sperm cells, or they're slightly different size or shape. So that's a pretty powerful identification tool, is to say, well, there's a sperm cell, so that means it's semen. Sperm cells will remain intact for a number of hours, depending upon the situation, but at least several hours. They will remain not only intact, but they will be swimming. Their tails will be flagellating. And so that needs to be recognized and noted, because if the time of the incident is in question, it may be helpful to say, well, not only did we find sperm cells, but they were still alive, or they were still flagellating, at least. So that's something that needs to be checked, and the doctor will check that at the same time as he's looking at the microscope. Or if not, then you should very rapidly get it to your own lab and examine it. It's a very, very simple examination. So if the stain is 15 or 20 hours old or older, then it will be dried out, and the sperm cells will all be stationary and not swimming anymore. And the first thing that happens in the decomposition process is that the tail falls off. So then you're left with a cell that looks like the head of a sperm, but could be confused for some other cells. So we have a stain that we use. We call it the Christmas tree stain, because it stains the sperm head red. Part of it's red and part of it's green. So we can stain that and look at it, and then we can identify a sperm cell, even though the tail is missing. And that we can do for a long, long time, as long as the sperm head is still intact, which, it's often many hours or even many days. I had one case where a body was not found for several weeks, and still able to find a intact sperm head on her clothing. So that's the preliminary testing, that is the testing that is done on the sample to examine for sperm. Semen has a component known as acid phosphatase, seminal acid phosphatase. There are lots of acid phosphatases. There are also alkaline phosphatases. They're enzymes in your body, and lots of parts of your body have acid phosphatase, but semen has it in concentrations much higher than other fluids and stains. So a positive test for acid phosphatase, although somewhat quantitative, is considered to be one of the really good tests to identify a dried stain as semen. There are two compounds which are used in the acid phosphatase test, one of which turns blue, the other which fluoresces. They both work and they both are adequate. And they can be used in the field, although typically they're not. They're typically used only in the laboratory. The next identification, if you get a positive acid phosphatase test, is a test for prostate-specific antigen, or PSA. PSA is a marker in semen that is produced in the prostate. That's why it's called prostate-specific antigen. And we now know that it's found in lower quantities in other parts of the body, but still higher quantity of PSA indicates that it is semen. It's also used by doctors as a test to determine or to give an indication whether a person has prostate cancer. It examines if the level of PSA in blood, and it is again much lower in blood than it is in semen. So prostate-specific antigen is tested for with an antibody-antigen reaction, somewhat similar to what we talked about with blood. We make antibodies against PSA, and then we can electrophoresis them, or we can test for them on other agar surfaces. Again, not a field test, but one that's conducted in the laboratory. So if we start off with a white crusty stain that fluoresces blue, and then we do the PSA, and we do the acid phosphatase, we consider it to be semen. And we could, of course, do antibody antigen for human semen. And there is a test using the same type of material that we used for blood. That is one of these antibody antigen hemotrace cards, only they're not called hemotrace cards. They're called PSA cards, or seminal fluid, and they work the same way. You dissolve a little of the stain and drop it on the end, and if you get 2 lines, then that verifies that it is human semen. So once we've identified the semen, we'll talk again more about the DNA of semen samples later on. I should note that it's a common misconception that the failure to find semen proves that a person was not the victim of sexual assault. In our laboratory, we found that in 30% or so of rape investigation kits, no semen was found. And that doesn't mean in 30% of the cases the victim was not being truthful. It just means there wasn't any semen found. And that can be for a number of reasons. The assailant may have worn some protection. This assailant may not have ejaculated for one reason or another, or ejaculated outside the victim. Any number of reasons, but we don't know which of those reasons is correct. All we know is we didn't find any semen, and we make it clear that that doesn't mean that the victim was not sexually assaulted. So I don't know that that's a well-known fact, but it is absolutely well known in the forensic laboratory situations that it's not uncommon to not find semen even though a rape occurred. On the other hand, finding semen also does not prove that sexual assault occurred. Obviously, the victim could have had consensual sex with someone else, and the semen could remain intact. We found an intact sperm cell in a young woman who'd been dead for 14 days, which at the time was a record, and I think it's subsequently been broken, but it's certainly expect to find semen up to 24 or 40 hours even, if, depending on the situation. And so it's obviously important to interview the victim and see if there's a possibility of some semen from a prior consensual event. And if so, then it will be necessary to get DNA samples from the previous partner or partners and eliminate them from the source of the semen that you find at the time of the rape examination. Once again, the presence or absence of semen doesn't in any way prove rape. Rape is a crime of consent or non-consent, and we can't determine from the physical evidence of finding semen whether there was consent or not consent. We can only determine that there's semen there, and if there isn't semen there, we don't know why it just wasn't found. Occasionally we find other stains. Saliva is relatively common as a result of biting or spitting or whatever. Saliva contains a number of enzymes. The ones that we're interested in are called amylases. Amylases are enzymes that convert starch to sugar, and you have strong amylase in your saliva. So we test for amylases. It turns out that we can use a very simple test in which sugars will change to starches, and the starches will turn iodine dark. And so we put in a petri dish, we put some agar that has starch in it, and then we cut a hole in the agar and add some of our solution of our sample, and it will diffuse out into the agar. If it has amylase in it, the amylase will turn the starch to sugar. So we wait a little while, and then we add some iodine. The iodine will change the color of the agar wherever there's any starch. If there's amylase, there won't be any starch because it will be changed to sugar. So that's a great little test for the presence of saliva. Not one you can use out in the field, but nice, fun little test to use in the lab. About the only other biological fluid that we find with any regularity is urine. Urine we do not have a good field test for, nor a good lab test that specifically identifies urine. But urine could have DNA in it. And so I always recommend that if you're searching a crime scene, a burglary scene, for example, you should check the toilet. If the water in the toilet is yellow, you should take a sample. You may be able to get DNA from that sample. So this concludes this movie. We're coming towards the end. Looks like I'll have to do one on DNA, and then a final one on arson and explosives. So thank you for your patience, and I certainly welcome any comments you have about these movies. I realize that I'm not a professional videographer, and I would much rather be standing in front of you in person, but we have to do the best we can under the circumstances. I hope you're all staying safe and staying indoors as much as possible, and practicing social distancing. I know we are, and so far we've not had any problems. So I hope you can report the same. I'll see you in the next movie.